Hello guys, today we'll discuss the enzyme based molecular diagnosis. So in our second module in the medical biotechnology subject, the enzyme based diagnosis is very much important topic, and according to our syllabus there are two enzymes that we need to study. One is glucose oxidase enzyme and the other is monoamine oxidase enzyme. In the glucose oxidase, so in first enzyme, that is glucose oxidase, the particularly glucose oxidase enzyme which is used to diagnose diabetes disease. So diabetes mellitus is an incurable disease. If you see, uh, because diabetes is a disease which occurs due to increase or decrease in the production of insulin in the patient. So insufficiency of insulin in the body causing elevated blood glucose level, which is known as hyperglycemia, and the decreased or reduced glucose concentration in the blood, which is known as hypoglycemia. Insulin is a hormone which is synthesized and secreted from the pancreas to mediate metabolic reactions which is involving glucose. In doing so, it initiates glucose uptake by the cell types, different types of cells in the body, and hence reducing glucose concentration in the blood. Diabetes has been associated with many medical conditions like celiac disease, cystic fibrosis, tuberculosis, heart disease, retinopathy leading to blindness, nephropathy gives rise to renal failure, peripheral nerve damage with increasing risk of like foot ulcers, cardiovascular diseases, and even though in worse condition it is the cancer. So diabetes has been described as a silent epidemic. In the plethora of the biosensors have been developed to provide diagnostic information regarding the patient's health status. So how exactly we monitor the glucose with the help of these enzymes? As a preventive treatment or a cure for diabetes is yet to be developed, guys. So still no any particular cure has been developed who treats diabetes. But at least we can monitor the blood glucose level in the patient to treat or to monitor glucose and maintain their health, diet, and although the exercise that patient has to do. So glucose monitoring can be used to optimize patient's treatment strategies, and also the although blood glucose monitoring is the gold standard medium for glucose sampling and measuring carried out in these fluid are invasive. So blood glucose concentration are typically in the range of 4.9 to 6.9 millimolar for healthy patients, increasing up to 40 millimolar in the diabetes after glucose intake. So this is like the sudden increase in glucose leads to any disorder in the patient's health. So get rid of this increase in the glucose level or to monitor these sensors were initially based on electrochemical approach. So electrochemical approach means what? The electrochemical conductors or the devices which is used the enzyme glucose oxidase. Electrochemical sensors were chosen for blood glucose measurement due to their rich or high sensitivity on order to micromolar to millimolar concentration, good reproducibility, and ease of fabrications at relatively low cost. So glucose oxidase was employed as the enzymatic basis for the sensor owed to its high sensitivity for glucose, guys. So glucose oxidase was employed as the enzymatic basis for the sensors, and which is having a high sensitivity. In the next slide, we'll discuss the other enzymes which is helpful to measure the glucose level in the blood, such as hexokinase or glucose 1 dehydrogenase, but they are less efficient or sensitive in contrast with glucose oxidase enzyme, which is which can tolerate extreme changes in the pH, temperature and ionic strength in comparison with the other enzymes. This is the indirect quantification of the glucose concentrations, and which is achieved by placing this enzyme glucose oxidase on the thin layer of platinum electrode via semi-permeable dialysis membrane. Semi-permeable dialysis membrane is coated on these electrode or these circuit, and this dialysis membrane is occupied or which is been having this glucose oxidase enzyme. This sensor measures the decrease in oxygen concentration and elevation in oxygen concentration also. So decrease in oxygen concentration measurement, the liberation of hydrogen peroxide, which was proportional to glucose concentration. So we'll see how exactly this enzyme works. Glucose is reacted with glucose oxidase enzyme, turns it into gluconolactone product, and the gluconolactone product is the mediator or intermediary product in the reaction. So we'll see how exactly it happens. Glucose oxidase catalyzes the oxidation of glucose to gluconolactone in the presence of oxygen, while producing hydrogen peroxide and water as a byproduct. Gluconolactone further undergoes the reaction with the water to produce the carboxylic acid product, that is gluconic acid. Glucose oxidase requires redox factor to carry out this oxidation process, so it uses FAD+. You can see here, that is flavin adenine dinucleotide is employed, and is an electron acceptor which become reduced to become a FADH2 during the redox reaction. The subsequent reaction with the oxygen to produce hydrogen peroxide, is it hydrogen peroxide, and which regenerates FAD plus cofactor. This reaction occurs at the anode, which is positive end of the electrode, positive electrode, where the number of transferred electrons can be correlated to the amount of hydrogen peroxide produced, and hence the concentration of glucose. So these reactions, at least you will understand how exactly glucose is measured, which is comparison with the hydrogen peroxide or the electron is liberated from the reaction. So this is how the circuit or the device which is used to measure the blood glucose level. You'll know that this is a small chip situated over here who contains that small circuit or platinum electrode, which is the covered by semi-permeable membrane, which is having glucose oxidase enzyme. So at least you will understand how exactly blood glucose level is being monitored through glucose oxidase enzyme. There are multi more applications are there, and this is actually the enzymes which is used to diagnose the diabetes. Thank you.